So we have started recording and the live telecast in YouTube. So just the live will start in a moment. So I would like to welcome everyone for today's talk. So this is, as you all know, this is part of a long series. This is called NASI Women in Science lecture series, and this has a history, means how it started. So I'll briefly tell, because many of you are regular visitor of this lecture or regular participant of this lecture. So but still there are some people who have joined for the first time, and so I would like to repeat that. When National Academy of Sciences India was established by Meghnad Shah, then he had an idea that to have something analogous to Royal Society in India. And one of the agenda was to connect science with society. And now, with time, there are three science academies in India: National Academy of Sciences India, NASI, Indian Academy of Science, and INSA. And they have different mandate. And mandate of NASI is to connect science with society. So last year, as part of 75 years of independence, when government approached National Academy of Sciences to organize several programs related to science but which have impact on society. So one of the particular program which NASI has decided to organize is a series of lecture called NASI Women in Science lecture series. At that time, Corona was already there and offline meetings were not possible. So it was planned that for the entire year, online lectures will be organized and the speakers will be eminent women scientists, and with a focus to encourage young PhD students, undergrads and graduate students to do mainstream science or continue science. And we have already organized 6 lectures, and this is the 7th lecture of the series. And without elaborating on the genesis of this lecture series, I would now like to request my colleague, Professor Papia Chaudhary, to introduce today's speaker.
Yeah, thank you, Mr. Pathak. Welcome you all on behalf of Department of Physics and Material Science and Engineering on this occasion that we are following that the lecture series. So today we are at 7th lecture, and we have with us Dr. Ankona Datta. So without wasting time, let me introduce Dr. Datta first. Dr. Ankona Datta is presently working as Associate Professor in Department of Chemical Sciences at Tata Institute of Fundamental Research, popularly known as TIFR Mumbai. Dr. Datta has received her graduation and master's degree in chemistry from the Indian Institute of Technology, Kharagpur in West Bengal. And after that, and her PhD degree, it is on tidal water soluble porphyrins for catalysis and recognition from Princeton University. After that, she worked as postdoctoral scholar at the University of California, Berkeley, where she worked on the macromolecular MRI contrast agents. And since 2010, she is a faculty in the Department of Chemical Sciences at TIFR. Her research interests are in the broad field of organic and inorganic chemistry, with an emphasis on chemical biology and molecular imaging. In TIFR, Dr. Datta's group, they proposes to use a range of tools pertaining for the aforementioned fields to explore new frontiers in disease detection and to understand the fundamental molecular mechanism underlying those diseases. Specifically, her group is interested in designing and applying novel chemical probes and sensors for imaging disease markers, both in vitro and in live biological samples. So I welcome you, Dr. Datta, to give your talk and enlighten us.
Okay, just give me a minute. I think this is just hanging. Now this is fine. Okay, thanks a lot, Dr. Chaudhary, for your very kind introduction. Are you able to hear me perfectly?
Yeah, we can hear you.
Okay, great. Today, I thought I'm not very familiar. I was not very familiar with the audience. So I just thought of putting things from the very basics and then to slightly advanced to cater to all the audience that we have today. So it's a pleasure to, be a part of this series. And today I'm going to tell you a little bit about our group's work at TIFR, what we have been up to. And so the title of the talk that I have given today is Smart Sensors Capture Life in Action. And by life in action, I literally mean looking inside biological systems, looking inside living systems and trying to see what the molecules are doing. So we'll start with a little bit of introduction, very basic introduction. And since I got the input from Anirban yesterday that, there are not too many questions sometimes. So I thought maybe I will also, ask questions as we go through the talk and we'll see how people are following. So you can please unmute yourself and answer questions as I pose them during the talk. We'll keep it very interactive. And so on the right side, what you are seeing over here, I'm hoping you're able to see the hand right over here. That is moving, my pointer. Are you able to see the pointer on my screen?
Yes, it is visible.
Yes, pointer is visible.
Okay, excellent. Okay, so on the right side, what you are seeing is the brain of a frog, and what you are seeing are different neurons, and you are seeing a calcium sensor inside the neuron that is basically sensing the calcium ions that are moving from one brain cell to the other, and this is how signals are transmitted in biological systems. So the key here is to understand that molecules inside our biological systems are the essence of signaling processes and all living processes that are going on within biological systems. So the question is, if we would like to understand how life works, then we would also want to track molecules within living systems. And so with that, I am going to begin my talk from the Tata Institute of Fundamental Research, Mumbai. And of course, we are still in the offline mode, and you can see a beautiful background of TIFR behind me. It's a very beautiful campus by the sea. So what did we start doing till 2010 is what I'm going to start telling you, but with very basic. So I said that we want to track the molecules of life. So the first question is, what are the molecules? And the molecules of life range from, of course, DNA, proteins. And so let us, gauge. So any student in the audience can tell me what molecule is this one? Anyone? I'm not sure if there's a chemistry student in the audience or not. So I'm just going to try and see if anybody can answer. Anyone at all? You can also write in the chat, so we will be able to see.
Student, don't be afraid. Means even if something is coming in mind, just
so
tell
I
me.
asked a question in the beginning of my talk.
Yeah. So I told you yesterday also, they're very reluctant to
No, it's okay. They are quiet. Okay, so anybody can guess what?
I think someone has written something.
Amino acid. Someone has written.
Excellent. Okay, this is an amino acid. This is tryptophan, and this is sugar, and this is water. So these are the essential molecules of life. I wish I, had some. If this was a live talk, I could have given chocolates or something so that people would have answered questions. Okay, so these are the molecules of life. There are many other molecules in biological systems that essentially work to make our, biological systems work. So essentially what happens is, these molecules, for example, proteins, catalyze reactions, and they can convert. For example, here you are seeing that there's water oxidation going on. There is reactive oxygen superoxide is getting converted to oxygen. Then there is the synthesis of some amino acids. All of these reactions form what is known as the chemistry of life. But it is very important also to understand that life works only when many molecules come together and interact, and they regulate processes, biological processes. So just like in the society, things don't work if people don't interact with each other. Similarly, if molecules do not interact with each other in a particular way in living systems, then you would not have life working. Okay, so so essentially this is what is critical. And if the molecules, for example, so what I did is I just put cartoons of people. It's not as if molecules look like people, but I'm just saying that if these molecules like people interact with each other, then you have, the process of life goes on really, really well. So molecular interactions drive life, and if things interact with each other, for example, calcium, sugar, protein, or whatever is interacting with each other, they are binding to signal a process, then you are going to be fine. Now, on the other hand, if molecules are not working, you get disease. So since time immemorial, we have tried to understand how life works. So, whether you're of a physics background or of biology or of chemistry background, we would like to understand what is happening in the nature around us. That is what scientists are trying to do for many, many decades, and eras have gone by when we are trying to understand what is going on around us. With a similar curiosity, chemists and biologists are trying to understand how molecules are behaving within the living system. Okay. So I hope everybody is with me so far. And if, I'm going to start calling names now if I can't get any response. But Shruti Sharma, I can see Shruti Sharma here. Shruti, are you a student?
Yes, ma'am. I'm a research scholar.
You are a research scholar. Shruti, are you, so which subject are you in? Shruti?
You can unmute yourself and speak.
Yeah, please go ahead.
Yes, ma'am.
So what is your subject? What are you doing your PhD on?
It's basically on quantum dots, like nanomaterials.
Okay, excellent. So you will relate to this. Okay, fine. So Shruti is doing her PhD on quantum dots. So essentially, what we are trying to do is we are trying to mimic nature. We are trying to understand what is going on around us. And one of the ways of doing this is basically taking the molecules of life and taking them in a test tube, isolating them, and then trying to study them spectroscopically. But there's a problem with that. And can somebody tell me what is the difference between just taking, so let's say I isolate proteins from a biological system, I isolate sodium, potassium, whatever is there, and put them in a test tube. Will I get a living system? Why don't I get a living system when I isolate the molecules, the same molecules that are there in my body, and put them in a test tube? Okay, I'm going to ask this to Anshu. What's name? Anshu, are you a student? Okay, Sukanya?
They are isolated, so they will not interact with each other, and hence they will not.
Well, they would still be able to bind to each other, right, Sukanya? Because let's say you have a protein and you have a substrate, it will still do the reaction, right? So if it does the reaction, then why don't I get a living entity over it? Okay, so fine. I mean, so I'll probably give you one of the answers to this. One of the major things with life is that molecules are not just moving by diffusion. Okay, what is happening is there is a particular and there's a distribution of molecules in the biological system. So it's obvious that if I take all the molecules that are there in my body and put them in a test tube, I still would not get a living system, right? So essentially what is going to happen is you have the molecules distributed in the body in a certain place, and the distribution where the molecules are is very critical to how we are sending signals. So in order for life to function, one entity of the biological system is a cell. So most of you must have read it in your 12th grade, or even like when you were in school, that the basic entity of life is a cell, right? So in order for one cell to interact with each other in multicellular organisms, signaling processes have to happen. And in order for these signaling processes to happen, essentially, the molecular distribution has to constantly change in a regulated manner. It can't randomly change, but it has to change in a regulated manner. And this kind of regulation I cannot achieve in a test tube. Is that clear, Sukanya?
Yes.
Excuse me, ma'am.
Sure, yeah.
Here, ma'am, I have a question. Does temperature play a role in this entire process?
Okay, so the body temperature, as you know, has to be maintained at a particular temperature so that the things are going to work. So local temperatures are not going to play too much of an effect in this process, because of course the body temperature, physiological temperature is going to be maintained at 37 degrees centigrade. But on the other hand, if you take the molecules of life and you would like to test to how their reaction rates, etc., would vary with temperature, you would definitely see an effect. But that would not be a physiological or a natural effect, right? So on the other hand, how do we sense temperature? How do we sense heat? Cold is also related to the signaling processes. Does that answer your question?
Yes, ma'am. Thank you.
Okay. Who was this?
This is Priya.
Oh, Priya, excellent. If I had a chocolate, you would have the chocolate for the first question asked. Okay,
Thank you,
excellent.
ma'am.
Okay, thank you. Please continue to ask questions. That's why I can understand whether you are following or not. Okay. So in essence, there are many ways. When we were in school, we read that, of course, living things reproduce, living things are able to, and all of that. But apart from that, from a molecular perspective, it is related to the fact that we are able to, the entire system that we have, even a single cellular organism is able to sense what is happening in its surroundings and give and transmit signals. Okay. And that is related to the location and position of molecules inside my body. That is very, very critical. Okay. So to explain this even better, as I was saying, why is this so important? Why am I thinking about this so much? The reason is because when the location and the distribution of molecules change, right? At that time, we have diseases. So for example, it is possible that increase in sugar levels in a person can lead to a certain disorder. Increase in metal levels or increase in some toxic metal like mercury or arsenic can lead to toxicity. So essentially, if molecules are not placed in the correct position, molecules are not behaving in the correct way, then we have diseases. This is basically the essence of all diseases that we have in the biological system. So disease conditions like neurodegeneration, cancer, inflammation, how do viruses infect us, what happens during these are all related to the molecular distributions within a biological system. So in order to understand how are these molecules behaving in the normal condition, basically to understand how life works, that is, the basis of life, we would want to visualize molecules and also to understand what went wrong. So, for example, it's very difficult for me right now to find out what each of you are thinking, what is your expression as you're listening to my talk, because I can't see any of you except for Anirban. Everybody's video is turned off, right? So I really don't know what is happening. It's a completely dark situation. Okay. So similarly, if I just, see something from somebody from outside and that person has a disease, I have no idea of what has gone wrong. Okay, so I would have to figure out what has gone wrong. And in order to figure out what has gone wrong, I have to look within. So let us take an analogy, a simple analogy of the Earth, right? So for example, you can take a satellite image of the Earth from outside and, try to figure out if something is going wrong in India. Would you be able to find out? It will be very difficult for you to do that, right? So similarly, when the doctor is looking at you from outside and, trying to figure out what is wrong, often it is very difficult for them to figure out what is wrong at the molecular level, leave alone what is wrong at the organ level, right? So just by looking at outside, they can give you something that, something is going on, some problems are there, whatever, but it's very difficult to say what is going on. So you would have to zoom in. So for example, here I have taken an image from Google Earth and we have zoomed into TIF. Okay, Tata is where I am right now. So this is what the doctor sometimes tries to do. They try to zoom inside, and what they do is, this is a very easy question, so I'll just answer this. So what they do is, they, for example, if something is wrong, they will take an anatomical image like an X-ray. So let's say somebody has broken their bone, you can take an X-ray and you can try to figure out, what is wrong. I mean, where did we have the fracture, etc., etc. Okay. So this is good, but still, in many cases, we do not understand what exactly happened. And so then you have to go to a diagnostic clinic. Sometimes they will measure the calcium levels. Sometimes they'll measure the sugar levels. Sometimes they'll measure lipid levels. They'll say lipid profiling. Even in the case of SARS-CoV-2, I think almost all of us have COVID by now. So essentially, all the different kinds of tests that were done, the protein tests and all of those were done to find out what is the level of molecules, right? So how did the viral infection basically affect the level of molecules in your body? But, that also does not tell, in many cases, the distribution of molecules. And finding the distribution of molecules can be very, very critical in certain diseases like cancer and neurodegeneration. So the question is, now this image that you have is just, going to be able to tell you a little bit more about what is going on in the biological system, but to actually elucidate the molecular mechanisms in biology, to diagnose pathological conditions, we will have to visualize the molecules. So in essence, I not only want to take an image, anatomical image of the organ or the bones, etc., I also want to see what the molecules are doing. But the problem here is that the molecular distributions and levels are not static. As I'm speaking to you, the molecules inside my body, inside my brain, they are moving from one location to another in seconds, milliseconds, minutes time scale. And that is how signals are being processed. And all of this motion inside my brain, inside my biological system, if it does not happen, imagine, millions and billions of molecules inside one biological system, even a single cell, and they all have to move in unison. Their distributions have to be in the correct position. The distribution has to be correct. Otherwise, the biological system is not going to work. So it's imagine so if you try to do this, you are trying to basically, you have to take images of the molecular, right? So the question is, what would you have to do? Let's say you took some still images of a biological system and for some method, you are able to see where the molecules are. But it is very difficult to just take still images at different time points and construct what has happened, right? So I just want to take a video of everything. So for example, imagine this World Cup match in 2019 within India and Pakistan, and I just give you 3 images. You have missed the match and because of an exam, and then the next day I give you 3 images. Can you tell me what has happened in this match in whatever sequence? Because I have just given you these images in random sequence, right? Looking at this seems like Pakistan is doing very well, but actually India had won this match, right? So essentially it is very difficult to figure out what is happening unless you are actually able to see the players and you are able to record events just like we are able to record matches live, right? So we need recording to visualize a series of events. We need to see what molecules are doing inside the biological system. So in order to visualize this chemistry within living systems, we have to realize we have 2 things. One is the object that I want to see. What do I want to see right now? I really desperately want to see your faces. So if you can turn on your videos, that would be great. But if any of you want to, but so right now I can see Sudha Srivastava, for example. I can see Papia. So essentially, the biomolecule, just like Sudha and Papia, has to be detectable, right? I need to see them. And I also need to see the camera. I also have to have a camera. So we have a camera, so I'm able to see you, which is going to basically have the resolution. Now, I'm able to see Dr. Papia Chaudhary, I'm able to see Dr. Srivastava, I'm able to see Dr. Pathak. But the thing is that is because the resolution is good enough to see them. But when we are coming to biomolecules, how small of an object do I need to detect is the first question, right? So, for example, if I want to detect a protein. Now, here is the question to the students, and I'm going to ask Satish. Satish, are you, awake? I hope you are. Satish Kumar? Yes.
Yes.
Okay, okay, Satish. Can you tell me how big are proteins? Doesn't matter if you don't know.
It is nano size.
Yes, excellent, excellent, excellent. So proteins are few nanometers in size. Now, so just to get an estimate of how big a protein is, if you look at the thickness of a paper, for example, it is about 100,000 nanometers. Okay, so you can see the 1 divided by 10 to the power 5 of this thickness of the paper is basically 1 nanometer. So we need to be able to see things that are nanometer dimensions. Fine. So now what kind of camera? Now the important thing is I want to visualize these biomolecules inside living systems. Now I can easily take pictures of you without doing anything to you, right? But the point is, if I try to take pictures of molecules inside a living system, I cannot kill the living system. It has to be alive, right? I want to take a video. We want that. So this means that whatever technique I'm going to be using to visualize has to be something that is non-invasive. What is the meaning of non-invasive? It means that essentially it should basically not hamper the biological process. I mean, the living system should be intact. And so the camera for doctors, for example, they do magnetic resonance imaging, MRI. So that is something that can tell you what is inside the brain. So you can do MRI. And this is, for example, inside the brain of a human being. This is inside the brain of a rat. And this can be used to diagnose anatomical abnormality. This is just telling you what is the relaxation of water protons inside the brain or in the biological system. But you can also look at molecules. For example, in positron emission tomography, you can see how a radioisotope or for example, fluorine 18 decays, and you can, for example, see the location of sugar that has been labeled with the radioisotope inside the brain. This is just an image of the brain, the cross-section of a brain, and the red portions is showing where there is high sugar uptake. Basically, they have inserted a radio-labeled sugar, which is now decaying, and it is giving you the images. But the problem here is that you can already see that the resolution is not such that if the brain is this big over here on the screen, which means that the resolution is not such that I can see individual molecules or even get a molecular distribution at a cellular level. That is going to be difficult using this technique, or even magnetic resonance imaging, which has a resolution of close to millimeter. So if we really want to look at molecules and what molecules are doing and how their distribution is changing at the cellular level, we need to zoom in further. So this is, for example, just a zoom in image using a fluorescence microscope of a slice of a brain where you're seeing multiple neurons. The small dots are neurons, and each of these neurons has been labeled with a dye that is a different color. So what you need to do is you need to look at all of these imaging techniques in unison, and you need to tell me now, for example, if you look at this plot here, right? So you see that fluorescence imaging is a technique where you have the resolution to go down to look at objects that are as small as 1 micrometers or sub-micrometers, right? And it has a time resolution where you can see, you can basically take images as fast as 1 millisecond, even nanoseconds these days, and going to several hours, which means that if I use fluorescence imaging, which is also a non-invasive imaging technique that can be used to image cellular objects, that can give me a resolution of microns and temporal resolution that is in a very large, right? On the other hand, if you look at magnetic resonance imaging or ultrasound, all of these you are familiar with the names because these are clinical imaging techniques. Positron emission tomography, it does not give you the depth penetration. Like, it gives you great depth penetration. For example, over here, as you can see, you can go down to a meter, so you can look inside the brain, but it does not give you the lateral resolution less than a millimeter. So it only goes down to sub-millimeter, right? Which means that you can't see objects, you can't distinguish between objects that are lesser than millimeter, that have a distance that is less than few millimeters. Okay, so far so good. Priya, is this okay? I mean, are you able to follow me?
Yes, ma'am.
Okay, fine. So this is good. So then what do we do? So the way we do is we actually go to micrometer resolution, and that is what is possible with a fluorescence microscope. Okay, so now under a microscope, you will be able to see cells. The cells are like, I think who had answered? Satish had answered previously, and he had answered correctly that the molecules are nanometer in size. So you can see that at least with fluorescence microscopy, we can see things that are micron resolution. And these days with super resolution microscopy, we can even go down to nanometer level resolution. But you can see that this is, for example, the outline of a biological cell. This is the nucleus. And so at least I should be able to see molecular distributions if I use fluorescence microscopy. And fluorescence microscopy is a non-invasive method. You're just shining visible light on these cells. And we'll come to the details of that in a bit, but it does not kill the cells. So I can basically zoom in and I will be able to see living systems. So now we are able to zoom in and take images. But you know, the title of this talk was smart sensors, right? So where are the sensors coming in and why do we need sensors? Now the reason is very simple. Right now I have zoomed in and I'm seeing cells. But I'm not seeing specific molecules inside these cells, right? These dark things are just some organelles inside the cell. So the question is, how do I see molecules? So I'll give you the analogy again. So right now we have zoomed in, let us say, to Tata Institute of Fundamental Research. Now, right now I'm giving this talk over here. I hope my students are in the lab doing something. So we need to know where are the molecular players of my group located currently. Similarly, just like I would like to see where the molecules are located, I need to know where my students are located right now. Okay. So we need to now specifically identify molecules like we identify people in this area. That is what we are trying to do. So for example, I can easily say that Shruti, Kunika, Rajashri, all these people are located right now in Mumbai, but and also so is Mitarupa, but I don't know where this guy is. Okay, so we try to figure out where this guy is, and we also want to find out where he is. So we find out that these people are all in the lab working hard. This guy has already finished his PhD and is relaxing in Kolkata right now before joining his postdoc. So this kind of information I need for my molecules. Okay, which molecule is where inside the biological system and what is the molecule doing, right? Is it doing its correct function? Or, is it relaxing? Is it in a relaxed mode? Or is it doing something aberrant is what I need to get the information. But the question is, how do I distinguish between these molecules? How do I specifically, try to see in the backdrop of fluorescence microscopy one molecule? So if these are molecular players, for example, proteins, sugars, calcium, can I put jerseys on them somehow? Right, so that I know which team they are working for. So that is what we will be trying to do. So how do we do that? We do that with something known as fluorescent sensors, which can now go inside the biological system and light up specific molecules. For example, here we have these cells where a sensor has gone and it has basically specifically detected a particular kind of lipids in the cell membrane, and it has lighted up the location. So you can see these blue colored things on the membrane. These are essentially the sensors, and they have lighted up certain lipids on the cell membrane. Now you can say, Ankona, you just gave us the end point. You told us that you can make images like this. How do we develop sensors? How do we actually design sensors in the lab is what we are going to try. And why did this point?
There is a question in chat room.
Sure. Let us go with the question. Yes. Can you read it for me? Because I will have to probably escape.
Okay. Shandip, you can unmute and speak now. So the question is how much the water solubility of organic pollutants can be detrimental for its identification with some of the sophisticated techniques, especially for what is the starting pollutant.
Okay, so I think the question is related to if you want to detect a pollutant in water, right? What is the lowest amount that we can detect? Who is asking this question? Sorry.
Sandeep Chokar is asking, and he has written that mic is
Sandeep, can you unmute yourself? Then we can probably get
he has written that his mic is not working.
Okay, Sandeep. So probably you're trying to say how much of an organic, of a pollutant we can detect. It depends on the sensitivity of the sensor that we design. And ideally, what we try to do is we try to design sensors such that they have a sensitivity so that they can detect the relevant amount of biomolecule or molecule that is there inside the biological system or a toxic species. So in this case, for example, I'm not detecting a toxic species in the biological system. What I'm detecting is a lipid that is already there in the biological system. Okay. And here I will also later on highlight that when the distribution of these lipids change, they change during disease conditions. And if we can identify how this lipid distribution is changing on the membrane, we can actually diagnose many kinds of disease. Okay, I hope that addresses your question. If not, you can continue to write in the chat and we will try to address more. Okay?
So I have a question. Are we going below the wavelength limit? Because
No.
visible light is
No. So this is basically all. This is just normal confocal microscopy. So we are bound by the diffraction limit, of course. So here, this scale bar is actually 10 micrometers or 20 microns. So we are actually looking at a cell which is currently around 30-40 microns, and we are not looking at single molecules over here. What we are looking at is clusters of molecules, right? So for example, this one is many thousands of molecules sensors together. But of course, there are many lipid molecules, and it's telling us that the distribution of the particular lipid in this particular cell is more on this side. There are some punctae on this side, and then there is some distribution in the somewhere inside the cells also, where probably the lipids are being synthesized, right? So this is actually the endoplasmic reticulum. So we are not looking at super resolution. This is just normal confocal. Does that answer your question? Okay, great.
Yes. Okay.
What is the dimension of the nucleus inside the cell?
Okay, so this cell has very large nucleus, as you can see. This nucleus is around 10 to 15 microns. So you can see that the cytoplasm is actually much less. These are human embryonic kidney cells. These have very large nucleus inside. So the entire nucleus is this entire region.
Okay.
Okay.
Ma'am, excuse me.
Yes.
These sensors are wavelength specific or they are tunable, like for any wavelength one can tune, or for particular wavelength one has to use particular sensor.
Okay. So basically, that's a very nice question, Priya. Actually, what we try to do is we try to make sensors that are going to be excited and emitting in the visible region, because when we excite under the microscope, you don't want to use UV light because that is going to be detrimental to the cell.
Yes.
Now, the wavelength specificity is actually very important. For example, if I want to detect two different kinds of molecules, I might want to make a sensor that is emitting in the red and another sensor that is emitting in the green, right? So that I can detect both the things together. So essentially, I have a control as a synthetic chemist to make a sensor, like you can make quantum dots that are going to emit in different regions. I can make a sensor that can in which I can tune the wavelength and I can decide which wavelength it is going to emit.
Okay, it is tunable. Thank you, ma'am.
It's tunable. Yes. Okay, great. So please keep on asking these questions because it tells me, if you are following or not. So now, these molecules, now let us go into how can we design these molecules. So these molecules are basically nothing but fluorescent dyes, okay? And when you excite these molecules, they emit in a certain region. And these, of course, have very nice applications, not only in, science, but also in these funky applications, like you can have funky fluorescent shoes and all of that. So now let us come to fluorescent molecules. So fluorescent molecules, we have small molecules, for example, like this is the structure of fluorescein. I think that somebody told me that she's working with quantum dots. So you can also have quantum dots. You can have proteins which are fluorescent. And the basic concept of fluorescence is that you excite a molecule, it goes from the ground electronic state to the excited state. There's a vibrational relaxation, and then you have an emission. And these are the only molecules that emit in the visible or, in some other wavelength, in form of light. Those are fluorescent molecules. So we have these fluorescent molecules, but how do we convert it into a molecular sensor? Because this is just a dye, right? This is just, if you shine light, it is going to be fluorescent. If I put it in the biological system, we don't know what it is going to detect, right? But I want it to detect a specific molecule. So how do I convert it into a sensor is the next question. So we take a fluorescent dye, for example. So let's say we have these molecules. One has a shape which is a circle, it's just random molecules. I've just drawn shapes over here. just to explain the concept. So if I am able to make a jigsaw puzzle-like molecule, which also has dye-like properties, such that it binds such that it binds to your molecule of interest, right? So initially it is not fluorescent. Only when it binds to your molecule of interest, you have designed it such that it becomes fluorescent when you shine light. Okay. So this is what we try to do in my group. We are trying to make these molecular sensors. So let me give you one example of a very basic molecular sensor that can be used for detecting pH. And this molecular sensor is fluorescein, and essentially it has got an ionization dependent fluorescence. So essentially you can see that in its anionic form, it has got a very high fluorescence emission, but when it is protonated, it has low fluorescence emission. Okay. So now I'll show you what happens if I take a solution of fluorescein and I add an acid to it. So based on this information, can you tell me what is going to happen if I take a solution? So this is fluorescein. We are shining light on it, and you can see it is emitting bright green. Now, what is going to happen if I add protons to this? Like if I add an acid to this. So let me ask this question to Sandeep Mishra. Sandeep, can you unmute yourself? Okay, Sandeep is
Yes,
not.
ma'am. Yes.
Yes. Okay, Sandeep. So based on this thing that I have shown you, can you tell me now what is going to happen when I add acid to this solution?
So it will become acidic, and
Yes, it will
the
become
pH will
acidic, and
increase.
then
pH will increase.
pH will decrease.
Oh yeah.
Yes,
Decrease.
but that is true. But what will you see?
More fluorescence.
More fluorescence or less fluorescence? Based on this.
Yeah. Less fluor.
Less
Yeah.
fluorescence. Exactly.
Yes,
So let us see. Let us do the experiment. So we do the experiment, and you're going to see what is going to happen. So we just don't worry. Something is happening in the background. So you see, Sandeep, this is what is happening. It completely got away. The fluorescence decreased, right? And you can see this person also had some molecules on the hand, like, that had fallen on the glass. So that also you were able to see. You are able to see now, right? So on the hand, but you can see that the fluorescence beautifully decreased. So this is a basic fluorescence sensor, pH sensor. Okay. So now this is what my group tries to do. We are going to try and explain a few of the things that we are trying to do in the next few slides. So what we are trying to do is we are trying to make these kind of fluorescent sensors in lab, where we want to detect metal ions, we want to detect lipids, we want to detect pH changes within things inside cells. And we are able to, these are different cells that in which we are able to see these kind of molecules. So now the next question that you might have is, okay, the fluorescein concept was fine. You are protonating, some conjugation is changing, the fluorescence is changing. But for example, if you went to the lab tomorrow and you wanted to design a fluorescent sensor, how would you go about designing a fluorescent sensor? Okay, so let us go with a very basic. I'm going to start with the project that we started in my group when we joined, and I like to use it because it's a very simple example. So what we wanted to detect is we wanted to detect metal ions. Okay, now why metal ions? So if you look at the periodic table, we have a lot of different kinds of elements inside our body. There's a lot of carbon, nitrogen, hydrogen, oxygen, phosphorus, sulfur, but we also have these trace metals which are very, very important for us. Also, there are toxic metals that enter our biological system, like there was one question, and cause toxicity. So we would want to detect those as well, right? So we would want to detect essential metals, we would want to detect toxic metals. And there are many metals in biological systems. We are obviously familiar with sodium and potassium, calcium, which is there in milk, and so we need to drink lots of milk. We also need iron, zinc, copper, manganese. All of these metals are very essential for us. So for example, iron is a part of hemoglobin, we already know. Copper is a part of hemocyanin, which is basically the oxygen carrier in these kind of shelled animals. And because of copper, their oxygen carrier protein is actually blue in color, because when it is bound to copper too. And we have iron, and here we have the red colored hemoglobin inside our biological system. So, but what we were interested in, so basically metals in biological system are very, very critical. They are important in signaling in the brain. Sodium and potassium maintain the membrane potential. Calcium is important for neuronal signals. They are important as antioxidants. And they are also important in catalysis. So when I started my group, one of the metals that I was very interested in detecting was manganese. And I'm going to tell you why we were so interested in detecting manganese. There were two reasons. One was a chemical challenge and the other was a biological challenge. There were two challenges, and that's why we were very interested in detecting. So why did we want to detect manganese? Well, manganese is present in very high concentrations in our brain, about millimolar concentrations. And it is very important for removal of reactive oxygen species. Okay, so reactive oxygen species are very detrimental to the functioning of our brain. There is increase in reactive oxygen species like superoxide radicals and other radicals during inflammation, neurodegeneration, even during viral infections. We have increase in reactive oxygen species, increase in inflammation. So manganese, both when it is inside a protein, also when it is in the form of salts, which is also there inside our cells, like bicarbonate, phosphate, citrate is also there, removes reactive oxygen species. So it is our natural anti-inflammatory element. It's very important in the biological system. But on the other hand, when people are coming to the toxic angle, when people in mining and welding are exposed to manganese containing fumes, manganese can accumulate in their brain. Similarly, in children, when there are mutations in manganese transporters that are supposed, because these are essential metals, we do not make them in our body. We get them through nutrients like food, etc. So when there is mutations, we get, excess manganese in the blood, brain, and liver, and this causes Parkinson's-like symptoms. So I'm sure people are familiar with Parkinson's disease. This is a disease that happens with age, and you have got memory loss, you have got balance dysfunction, and all of these problems. So essentially, manganese exposure and excess manganese causes also Parkinson's-like symptoms. And this disease is very distinct from Parkinsonism. It cannot be treated in the same way, but its symptoms are absolutely similar. So from outside, any doctor would not be able to say whether you have got Parkinson's or enhanced manganese concentration. So what we felt is, we would want to understand, first of all, this metal is very important for the functioning of the brain. What is its role? How is it regulated? And because it's so essential, all of a sudden, if you get extra, why are we getting these diseases? So we wanted to locate this metal in cellular levels, what is happening in the cell, also in model organisms, because we can't do all experiment in human beings. So we took, organisms which can be imaged in a microscope also have a brain. And finally, we also wanted to figure out if there can be methods in which this disease can be diagnosed and therapeutics can be developed. So central to this, we felt was what we needed is we need to detect this metal ion. What is it doing inside the cell? Where is its location? And how does its location change during disease condition? So what we need is selective sensor, because we have many metal ions inside the biological system. How do you now? So all these molecules can be players. So calcium is there, zinc is there, proton is there, manganese is there, sodium, potassium, everything is there. How do I selectively make a sensor for manganese? The sensor has to enter the cells, has to detect the manganese, and then it has to detect the manganese at the levels that is there in the cells, that is micromolar levels, and also in the pathological conditions. Hopefully, it is going to be able to also detect the distribution of manganese in different regions of the cell, that is in different organs. So this was a biological challenge. Okay, so we want to detect manganese because it is important. Also, it causes diseases. So we want to understand what is going on. But the chemistry challenge there was this. And the chemistry challenge here is that manganese, basically, in the +2 oxidation state, it is a D5 system. So this is what you have probably read earlier. So it's a D5 system. At a high spin, it is D5, 5 unpaired electrons. Okay. Now, if you remember your basic chemistry, you will remember that the crystal field stabilization energy for a D5 system, because it's in the center of the transition series is 0, right? So if you try to make a ligand that is going to bind to manganese, manganese doesn't care, so it doesn't bind to anything strongly. On the other hand, the other metals that are there, they have crystal field stabilization, so they prefer either tetrahedral or octahedral geometry. So that is the basic challenge. It is very difficult to make something that is going to selectively bind to manganese over the other metal ions. So that is why this Irving-Williams series kind of said that it's going to be almost impossible to make molecules that will specifically and selectively bind to manganese. That was the challenge. So we took up this challenge and we needed something that is going to specifically bind to manganese and is going to become fluorescent. So we need a binder first, then we will think about fluorescence. So how will we do this? So we thought that how do we go about it? So again, basic chemistry, manganese is electron deficient. So first of all, a binder has to have electron-rich donor. So we will bring in nitrogen, oxygen or something that is going to be able to donate electrons to the manganese. Now, manganese doesn't prefer any geometry. So, but we know that other metals have a preference. So essentially, if I find a geometry which is not preferred by the other metals, then manganese doesn't care, it is going to still bind, the others will not be happy. So this is the kind of indirect approach that we thought to solve it. So zinc likes tetrahedral, iron likes octahedral, copper likes, again octahedral, but with square planar with, the axial ligands which are longer. So manganese doesn't care. It's okay with like this 5 donor atoms in a plane, which nothing like copper and other metals, they don't like it. So this is the idea that we actually used. Indirect method, when manganese will still bind, others don't want to bind, so they are not going to bind, and we are going to be left with something that can bind manganese. So this concept we use. The next thing is binder you have. Now we will have to bring in this turn on thing, right? So I hope people are following. Priya, Sandeep, are you able to follow? Sukanya? Okay,
Yes,
so,
yes.
so far. Okay, great. So let us go ahead. Okay. So now what we will do is we will make the molecule. So now let us come to the actual molecule. So you have these 5 nitrogens. Now the size of this ring is such that it matches that of manganese. Okay. Now also we realize that manganese is, its electron density versus its size is such that it's neither too hard nor too soft. So we use 2 oxygen donors also to eliminate certain metals. Like zinc can also be happy with 4 of these. So even if it does not use all of the donors, it can use some and still it can bind. So to eliminate some, we use some oxygen metals. Now the next thing is, how do I make this light bulb glow? So in order to make the light bulb glow, I will need to turn off the bulb first. So I attach a fluorescent dye, which I'm not drawing over here yet, I'll show you. But this is just a Bodipy dye. And what we did is, we put a dimethyl. So the linker that we used to attach is a dimethyl aniline. Now dimethyl aniline is a very well-known quencher of fluorescence by this process known as photo-induced electron transfer. Okay. So we shine light and this electron transfer actually quenches the process. Now, when manganese, this was our design. So when manganese is going to bind, it is basically going to use the lone pair of electrons. It's basically stabilizing the orbital, the electron pair over here. And so this electron pair is no longer available for quenching and the fluorescent dye turns on. So this was the concept. Okay. So now, of course, this is the concept, but there are certain people who have actually made this happen in the lab. It was not so easy. We synthesized this molecule, and the people who made this were my 2 first PhD students, Shubha and Anindita. Shubha is now a postdoc in Japan. She's a JSPS fellow, and Anindita is now a postdoc in University of Michigan at Ann Arbor. So students will relate to this, that, there's an end to the PhD or whatever you're doing, and you go to the next level, which is a postdoc. And of course, this is the molecule then, and this has a binding constant with manganese of around 4 micromolar. So essentially, you can see now this is a very rare molecule. In the absence of manganese, this is off, and in the presence of manganese, this is turning on, right? So this is fluorescence. Now, if you just look at the fluorescence intensity, how much increase you observe with different metal ions, you see that it is maximum for manganese and other metals are not responding. Zinc and copper are also responding to some. But the good thing here was that free zinc and copper inside our cells is very, very low, almost, 1 atom per cell, free zinc, it's mostly bound. So in essence, we would not get too much of, I would say, crosstalk of the sensor or, signals from zinc and copper. So ideally, we have the major signal for manganese. And this is a very rare sensor for manganese. So the next thing that we wanted to do is, if we have the sensor, great. Can this enter cells? And if this can enter cells, can this tell me the location of metals inside cells and also tell me what is happening under disease conditions? Okay. So we took this into cells, and you can see this is a cell, and you can see there is some bright fluorescence. This is the center, is the nucleus again, in a particular region inside the cell. Okay. Now, this particular region, what is this region and why is this accumulating the manganese was the question. Okay. It doesn't seem to be distributed all over the cell. It is mostly distributed in this particular region of the cell. So this is what Shiny, who is now a postdoc in MIT, this is wrong over here. She is in MIT now, and Kostav, who was an MSc student, he is in Texas A&M, USA. So they tried to enter. And what they were able to find is, I'm going to just give you the results, because the experiments that we did were in a synchrotron and they were very complicated, but I'm going to now reduce this to the school level. Okay. So this is the thing that we used to make in school. All of you have made this, the model of a cell. Okay. So what we found out with many experiments is that, so manganese, actually this region is the Golgi apparatus. Okay. And the Golgi apparatus is essentially the location which is responsible for removing metal ions from, or excess metal ions and excess toxicants from the biological system. So in normal human beings, what happens is we eat and we have essential nutrients like manganese. If there is excess, it goes out from the Golgi body. So we are fine. But in certain people, there is mutations in transporters. And what happens is that manganese stays inside the cytoplasm, and excess manganese cannot leave from the Golgi body because the transporters in the Golgi body are affected. This is what we found by using this sensor. And what this told us is that in case you have mutations in proteins that are able to remove manganese from the Golgi body, you would get excess retention of manganese inside your biological system, leading to diseases. So these are the kind of questions that we can now address. For example, how does the manganese location change during disease? So since then, what has happened is many people who are working on, so manganese is very important for immunity, for our immunity against viruses. So we have been getting lots of people who are contacting us, trying to use this sensor, for example, to see how the localization of this metal is affected when viruses infect our body, when we have cancers. For example, in lung cancer, it seems I got a recent email from a doctor in US who is saying that in lung cancer, the levels of manganese and a manganese transporter gets affected. So they want to use our sensor to see how the levels are getting affected. I said that manganese is very important for our defense against our defense against microorganisms and many other conditions. So in disease conditions, the disease factors try to utilize this metal ion and they try to, get an upper hand over this. So this is what my current students, Shruti, Punika, Shujit, and Lakshman and Chiranjit are trying to do. And they are trying to basically make these molecules such that they can use them for these different applications. So we are trying to now, in collaboration with people, utilize this sensor and second generation sensor for these purposes. Okay, so now I'm going to switch gears a little bit and tell you about one more story, maybe for another 10 minutes. Do I have another 10 minutes or so? We can go to another story.
Yes, you can.
Okay, so, this is the concept where I have a bulb that is off. I'm turning it on. But, this Diwali light kind of thing can also be done, where I have a sensor that has an emission in a certain wavelength and is going to emit in a different way. The advantage of this is that I can now quantify the level of a molecule because I will know the concentration of the unbound sensor. I will know the concentration of the bound sensor, and from the binding affinity, I can actually figure out how much concentration of this molecule I have. So that is how we can do quantification. So here so these kind of sensors, the previous one was known as a turn-on sensor. These kind of sensors are known as a ratiometric sensor, because now we are seeing a change in the emission. So here what we were trying to do is we wanted to detect lipids. So the question is, why all of a sudden do we want to detect lipids? What are lipids? So lipids are these molecules which have a polar head group attached to some fatty acid tails. Okay. So this part is polar, this part is non-polar. Now, because this molecule has a polar and a non-polar side, these things self-assemble such that the non-polar side looks towards itself and the polar side looks on the outside. And this is the essential molecule that makes the membrane of all cells that we have. Okay. So this is basically our cell membrane. So the essential constituent of our cell membrane is this lipid bilayer. Okay. So this lipid bilayer, of course, is a structural entity that is basically encapsulating everything that is there inside the cell. So the next question, I hope there are there any questions in the chat that we are missing, or we are?
No, not at the moment.
Not now.
Yeah, no, I have a question. So I want to ask, do the sensors always perform the qualitative analysis or the quantitative analysis too?
Excellent. And this is what we were getting at, that, you know, if we have a on-off sensor, it is difficult to do quantification. But if I have a ratiometric sensor like this one, right? So I will know the amount. So for example, basic chemistry, you have a molecule that is binding to another, you can find out the binding constant. If I know the binding constant, then if I know the amount of the unbound molecule and the bound entity, then I can figure out the concentration of this red species. So the ratiometric sensors allow us to do absolute quantification of the concentration of the molecules inside the biological system. The on-off ones do not. And that is why this ratiometric sensing is, you know, advantageous. Does that answer your question?
Yes, thanks.
Okay, great. Okay, so here in the lipids, it is important to do quantification and amounts, because, you know, when you go to a doctor, you do something known as lipid profiling often, like, you know, what is the level of lipids, etc. And the level of lipids and the kind of lipids that you have tell whether your health is okay or you are having some disease or some other condition. So essentially, this lipid bilayer that we have inside every cell of our body, unicellular organisms, multicellular, is encapsulating what is there inside the cell. But one thing that is not very intuitive is the fact that these lipids, now I'm drawing them as these red circles and these tails, because this is the polar head group and this is the fatty acid. Okay, and this is the cell membrane, and we have about thousands and thousands of variations of these lipids in each of our cells. So imagine the diversity. In every cell, we have got so many different kinds of lipids. And I'm drawing these as the polar head groups and these are the fatty acid tails. So why do we have so many variations if, you know, it's just a structural entity? The reason is because these lipid molecules actively participate in transmitting signals and regulating cellular processes. And how do they do that? What is the concept? Well, the idea is, let us say any information is obtained at the border of the cell. So let's say, you know, something is happening in the border of a country, some problem is happening. How do you know, like, how does Delhi get, or, you know, how does Calcutta find out about it? That is the question. So now I have a signal over here, some problem is going on. I need to block this position. What will happen? I need to send some proteins that are going to go there and block this signal or transmit this signal. But the cell, the first question that I had, somebody answered that the proteins are nanometer inside. Also from the images, you saw that the cell is micrometer inside. Now imagine how much do these proteins need to walk, or, you know, how will they get to this membrane? How will they know that something has happened few micrometers? So this 10 micrometer is 10,000 nanometers. How are these two proteins going to reach? So essentially what happens is many of these proteins have binding sites where they can recognize a particular lipid. So when a signal comes, often the distribution of the lipid on the membrane is going to change, and it's going to change such that, let us say, these proteins can bind to the green lipid. So what is going to happen? They are going to go and bind to the green lipid. As a result, this cell is a three-dimensional object. This three-dimensional search is going to get reduced to a certain location as a two-dimensional search in a very small area on the membrane, right? And the signal is going to transmit. So this is, of course, a hypothesis, and there are some evidences that these kind of things are happening. But what we would like to do is, we would, just like I'm saying, I would like to see these. I would like to see this distribution change, because all essential signaling processes in our body are happening via the distribution changes of lipids. And this distribution change, this intricate dance of lipids, is exactly what is basically transmitting signals, deciding whether we are healthy or not, also deciding, if the lipid distribution is different, then you know that you have a disease condition. So if we can detect these, then we can actually tell about what is happening inside the cell. Essentially, we can kind of map all these signaling processes. So it's very important to detect. But how will we detect? So remember, for the metal, we needed a binding partner. For a lipid, we already have binding partners, because I told you that the proteins are going to bind to these lipids. But the problem is, if I try to make a sensor out of an entire protein, a protein is, it's not able to enter the cell by itself. And many of these lipids are, you can see that there are two layers of the cell membrane. One is the outer layer and other is the inner layer, right? And many of the signaling lipids actually reside on the inner side of the cell membrane, because that is where the signal is getting transmitted from. So I need something that is cell-permeable. So what we did is, we said, okay, here we already know that a protein will have a binding site. So if I can just look at the binding site, and instead of the protein, because the protein is made of amino acids, just take a sequence or a portion of the protein that is made of amino acids, I already have the binding scaffold there. Okay, and we can easily make peptides in the lab. Next question is, again, the on-off signal. Remember, in the manganese case, we use photo-induced electron transfer. So now, how am I going to convert this peptide? Can come and bind with a particular lipid, but how am I going to convert it to the sensor? That is the question. So I put a fluorescent dye. But why will the fluorescent dye change its emission when it binds to the lipid, was the question. And this is what Shangshu Johar, my PhD student, addressed. Very simple concept. He's our postdoc in UPenn. And what the concept was, there are certain dyes which can change their emission when the dielectric constant of the medium changes. I think everybody is familiar with dielectric constant, right? The dielectric constant of water is 80, and the dielectric constant near this lipid region is close to 3. Okay. So if I take such a dye and I have this binding scaffold, the binding scaffold is going to specifically go and bind to a particular lipid on the membrane. Now, when this binding scaffold comes and binds, as a result, the dye will also come close to the membrane. When the dye comes close to the membrane, its emission is going to change, because the dielectric constant is going to change. So I will be able to detect specifically. Okay. Is that clear so far? So Shruti, was that clear?
Yes, ma'am.
Did you understand the concept? So this is a very simple concept. And the advantage is, many of these peptides are going to be self-binding. So just to show you this, if you take this kind of a dye in water, for example, it is emitting in the green. Now let us see what is going to happen after some time if I add an organic solvent of lower dielectric constant. It immediately turns into this. It's instantaneous. Okay. So if I can now use this very simple concept, I should be able to make a sensor. So this is what we did. We designed the peptide. It was not as simple as I'm saying, but designed a peptide. We took a ratiometric polarity sensitive dye. This is also excitable by 2-photon excitation, which allows us to excite at longer wavelengths. So we can actually look at these things in the biological system without affecting the biological system at all. And what we did first is, we made, artificial cells, so artificial membranes. So we are making these small unilaminar vesicles, which are 200 nanometer in diameter, and we are taking the specific signaling lipid. In this case, this is a phosphoinositide in the major phospholipid, that is phosphatidylcholine, and we made these artificial vesicles. Now let us see what we are seeing. So initially, this molecule was emitting in the green. Okay. Now, when I'm titrating in more and more of the lipid, I see a beautiful blue shift and an enhancement in the emission in the blue. Okay. Now, if I take the ratio of this blue to green and I do this experiment for all the lipids that are there in the biological system, I see that this is responding only to two kinds of lipids, which are known as the phosphoinositides. And these are actually key signaling lipids. I won't go into too much of the biology, but this class of lipids is actually involved in cancer metastasis. One of these lipids, when its concentration increases, it actually leads to increased cell growth and proliferation and movement. So it is very important to detect these lipids. So now what happens is, I take this sensor, I put it in cells. Okay. So what should I observe? Wherever there is water, it should be green. Wherever there is lipid, it should be blue. Okay. Because it's going to bind to that particular lipid on the membrane, it's going to give me an emission in the blue. So this is a three-dimensional intensity plot now, where I have added the sensor. You can see outside the cell, and the mountains is basically increasing, basically giving me the intensity. Okay. So essentially, you can see outside the cell is green. The sensor has entered the cell, inside is also green. Certain regions where there's high concentration of this lipid, you can see there's very high concentration here. The mountain is high, it's blue. This region is low in the lipid. You can see this is mostly there. So using this, I can now specifically, and the biggest advantage of this is this molecule automatically enters. So this is a cell permeable ratiometric sensor that can quantify the lipid. And we are now using this to basically, we are able to now take videos. This is just a snapshot. We are able to take the videos of the signaling process as it is happening, right? And we are able to see how the signal changes. For example, when we have a drug that is used to treat a disorder, how does the signal change? What happens when people get people utilize, for example, if somebody takes LSD, right? How does the signal change? So these kind of things we are able to actually see live, like the signal patterns change. Okay, this is what we are able to do. So what I was able to show two examples. One is metal sensing and the other is lipid sensing from very basic chemistry concepts, how we are able to utilize it for fundamental understanding of biological processes. But these kind of sensors actually have a lot of applications in disease diagnosis, pollution control, drug screening, detection. Of course, you have these detection kits now for the viruses, etc. So many of these are based on these kind of sensors. All of them are active. So, for example, one thing that a serendipitous discovery in our lab was a Mercury sensor. And you can see that this is the sensor which is visible now. Colorimetric sensor, which is pink in color. This is all real time, and you can see a solution containing a toxicant like mercury. This is all just water soluble, so we can very quickly detect mercury, like mercury toxicity. So essentially, mercury toxicity is a big problem in West Bengal because it makes its way through our body through fish, where mercury accumulates. And this is a sensor that is a rapid reversible sensor. As soon as we add a solution containing, it can detect 10 ppb, parts per billion of mercury. And it's a very sensitive molecule that can detect this. And you can see this as soon as I'm adding this solution, it is immediately changing its color, right? So in essence, these can all have many applications. And this is what our group has been doing. I could not tell you, I thought it's better not to tell you about all the projects, but actually take you through the process of how we design and develop sensors. So before we end, let me tell you a little bit about my journey as a scientist, the fun part actually. So in 2010, as Dr. Chaudhary mentioned, we began our lab in TIFR, and this was an empty room which we built into a lab. And it is now filled with many students. And, this is what keeps us going. This is the group. This was a group couple of years ago before the pandemic. And, but, just to tell you a little bit more about my journey, I, of course, did not start from TIFR. So my life started long back. And of course, there are questions like students often face, what do you want to do? Where are we going with this? And so on and so forth. So I started, as a kid in IIT Kharagpur. And apart from climbing trees, of course, I wanted to do many things. I wanted to become a singer. I wanted to be a police officer. Then I wanted to become a journalist. So many things I wanted to do. And the reason was because, they were role models. Because, I heard some Firoza Begum saying, Kiran Bedi was very popular at that time. Then I saw Nalini saying, and I wanted to do all these things. But at the back, so I'm chemist now, of course, and what gets me going is because I really love the things that I do, really love interacting with students, the wonderful people. And I'm sure Anirban Papia and other professors here would resonate with that it does keep us young and happy. But, also I had another role model. Only realized that later on, because in our CBSE textbook, some of you might, I don't know, you might have read, or I don't know, still has it. Actually, the CBSE textbook still has this thing. So we had this excerpt about Marie Curie. And this was, I think, class 5 or 4 it had. And, this is what I don't know why I used to like this excerpt a lot. And I read it on my own and I really liked it. And maybe I just read it for you. And this actually summarizes what it is to be a scientist, right? And since this is about women scientists, so what better than Marie Curie and what this says. So I read it in Hindi only because I'm sure everybody understands Hindi in this audience. So this is the chapter 10, Madam Curie. So there's an excerpt from here. सन 1902 की एक रात्रि को वह महान क्षण आ गया जब क्यूरी दंपति को उनके परिश्रम का फल मिलने वाला था। दिन भर प्रयोगशाला में काम करके, दिस इज आई थिंक आवर लाइफ, वे घर गए और बच्ची को खिला पिलाकर सुलाने के बाद बिना आराम किए ही घर काम पर वापस आ गया। सो दे पुट देयर डॉटर टू स्लीप एंड दे केम बैक एंड उन्होंने खिड़की से प्रयोगशाला में एक निगाह डाली तो वे स्तब्ध रह गए। अंदर अंधेरे में मेज पर रखे प्यालों से मंद मंद नीले प्रकाश की किरणें फूट रही थी। दिस इज रेडियम, आई थिंक। वे इस तरह खड़े खड़े उस प्रकाश को देखते रहे मानो उन पर जादू सा कर दिया गया हो। प्रोफेसर क्यूरी ने धीरे धीरे किवाड़ खोले और आश्चर्य तथा उत्सुकता से भरे वे दोनों अंदर घुसे। कमरे में मंद मंद नीला प्रकाश फैला हुआ था। मरिया खुशी से उछल पड़ी और कुछ ही देर बाद मरिया बोली, आह कैसा अद्भुत प्रकाश है, अंत में हमने इसे पा ही लिया। सो बेसिकली whatever level we are doing research, the ultimate goal, when you actually see the molecule works, or you have been trying to get this result for a really long time during your PhD, or it's not working, and finally when you actually get it, it's really that amazing feeling that nobody can take away from you, right? So a scientist might be very, I would say people who are not very glamorous or anything, but this is something that we have, that nobody can take away from us. When we see that spectra, and finally, wow, it is like there, right? So that is that amazing feeling. So what I would like to end with is that our country needs more scientists. So you will be the next role model. So we have a lot of hope from you all students. And with this, I'm going to end my talk and we can have more discussions and questions during the discussion session.
So, Vince, although you people have asked questions in between and some people have left in between, but still, if you have questions, please ask. And this wonderful talk will be also in our YouTube channel, means the shared link, and you can see it in future. And all the friends who have missed, you can tell them to.
Yeah, some of them are here still who had asked questions, and some of them.
So please ask questions, means if any.
Are there any other questions?
Okay, I have a very simple question. Like,
Yes.
like when we were talking about fluorescence in the brain and we're detecting manganese. So how do we detect that fluorescence? Like, do we
okay,
test
so we
this
cannot
in the lab
detect
or
it
in the body?
Yeah, inside the brain is very difficult because, as you know, fluorescence you can't detect it within below a few millimeters of distance, right? So essentially it has a certain, as I said, the depth penetration is not very good. So with fluorescence, you would have to detect it only, if you have to detect manganese, you would have to detect only in cells or organisms that are optically transparent, like zebrafish larvae, brain, and other things we can do. But if you want to detect manganese in the brain of a human being, which is also one of the sensors that we have made, we have to make what is known as a magnetic resonance imaging or any other kind of sensor, which can be used in these kind of imaging modalities, like, clinical imaging modality. So that is how you would do it. But in the microscope, of course, you can't use a microscope to detect manganese inside the brain. You can use it in cells, isolated cells, cultured cells, and so on and so on. Does that answer your question, Ashwin?
Yes, thank you.
I have a small question. With which wavelength you are exciting? The cubates you have shown there, that the green color. So what is the color you are exciting that?
Okay, so the excitation wavelength for the first manganese sensor is 480 nanometers.
Okay.
And for the ratiometric sensor, the excitation wavelength is actually 405 nanometers. But we can also do multi-photon excitation at 780, 800 nanometers. So that is why, that becomes more easier to deal with biological samples because it does not harm the biological sample.
Okay.
Any other question? If not,
I think we have. It's good that we asked questions in between and we had the question.
If not, we would like to thank everyone who are especially in Noida and here because the weather is very bad outside and still who are there. I would like to thank them to be here instead of a very bad weather outside. And I would like to thank the speaker for a wonderful talk. And again, I expect that many more people will see it in the YouTube, and this will serve the purpose. And especially the end part was very nice. That was in line with the way that the series of lecture was planned. And thank you very much for making everything so lucid and without using equations.
I felt that it would be nice, because the audience is very heterogeneous.
Thank you, and we'll close the meeting here.
Okay. Thank you. It was very nice meeting both of you. Bye-bye.
Thank you.